Welcome to part 2 of the immune system. In part 1, we covered the innate immune system, which would include the physical and chemical barriers as well as the non specific immunity. Under the physical and chemical barriers, we looked at skin and the different chemical secretions like sweat, mucus, tears. And then under the non-specific immunity, we looked at different types of white blood cells. So in part 2, we will concentrate on specific immunity or acquired immunity. Specific immunity is a type of immunity that we developed after we are exposed to the pathogens. And the two ways we can acquire it, one is by natural exposure to the pathogen, and then we become sick with the disease and we develop specific immunity towards that pathogen, or we could be artificially exposed to the pathogen through vaccinations. Right now, with the coronavirus, we don't have immunity towards it, and therefore the social distancing becomes very important. So, in some cases, some people may be infected with the virus but may not show symptoms, and they could unknowingly transfer it to another person. So, that is why social distancing is important in these times. So, some terms that you need to be familiar with is antigen and antibodies. So, antigen is any molecule, any foreign molecule that can trigger an immune response in our body. So, it could be any pathogen, any bacteria, any virus, but it could also be pollen or dust or anything that our body considers foreign. And what it does is, when it recognizes something as a foreign body, it makes antibodies towards that particular pathogen. Let's take a look at vaccines. So, what is a vaccine? So, vaccine is something that can contain the whole pathogen, or it can contain just a subunit, parts of the pathogen, or we could just make a DNA vaccine based on the microbe's genetic material. So, when a vaccine has the entire or whole pathogen, you could have it in two forms. The pathogen could be inactivated, or it could be in a weakened form, which is called the attenuated form. The subunit vaccine will only contain the essential antigens, so only parts of the pathogen. And so basically, the chances of adverse reaction to a vaccine having subunits is very low. And in the case of the synthetic DNA, the vaccine is made using the microbe's genetic material. So in this case, only copies of a few genes are present in the DNA, and those genes code for important antigens. So once we are exposed to the DNA vaccine, the DNA will enter our cells, and then our cells will make those antigens. And then the body will react to the antigens made by making antibodies. So, and DNA vaccine is very easy to make, and it's also inexpensive. So, looking at the vaccine that contains the inactivated and the attenuated form, there are some advantages to using one over the other. So, if the vaccine contains the inactivated form, it is not going to mutate into a virulent form. In other words, you will not get the disease from that particular vaccine. So, if a vaccine contains the live attenuated form, Sometimes, very rarely, it could mutate into a virulent form. And if that happens, we could get sick from the vaccine. But the chances of it mutating to a virulent form is very low. The other difference between inactivated and attenuated vaccines are the inactivated vaccines, they would produce a weaker immunity as compared to the immunity obtained from a vaccine having the weakened form of pathogen. So, an English doctor by the name of Edward Jenner first came up with the smallpox vaccine, and because of this, smallpox has been eradicated from the world. Another disease that is close to eradication is polio. Three more countries still have cases of polio sporadically, which would be Afghanistan, Pakistan, and Nigeria. I want you to think about this question. Can you get the flu from the flu shot? You cannot get the flu from the flu shot simply because it contains the inactivated form. Sometimes some individuals develop mild flu like symptoms, but you cannot get the full blown version of the flu. Um, if you get the nasal spray form of vaccine, it does contain the live virus, and so it might produce a stronger immune response, and it is not recommended for children and older people. Also, we have to get a flu shot every year because you can see from this slide that the proteins on the surface of the virus changes from season to season. So the flu virus undergoes mutation a lot, and each time it mutates, the proteins on the surface changes, and essentially a new strain of flu virus comes about, and we don't have the immunity towards the new strain, and that is why we would have to get a flu shot every season so that we can have immunity towards that particular type of flu that is present in a particular season. So, speaking of vaccines for coronavirus, we don't have a vaccine yet. It might take 12 to 18 months to come up with a vaccine for the coronavirus. Once they discover the vaccine, it has to be tested in clinical trials, both for efficacy and for safety reasons, and then it will be made available to the public. So, till we have a vaccine or some effective treatment, we have to be really careful with social distancing. Taking a closer look at the antibody, it is a Y-shaped, and these are proteins that are found in the blood plasma, and essentially they are made up of 4 polypeptide chains, and they can fold into different shapes, and at the very tip of the antibody you can see they can form bumps or grooves or different folds that will attach to a particular antigen. So antibody is something that is going to bind to the antigen, and so it is a protein that is made in our body. Here you can see antibody A with a different shape at the tip, and antibody B has a different shape, and accordingly they will bind to different antigen. So the antigen would be a virus or bacteria or any foreign substance. So how does the antibody protect us, right? So as long as the pathogen is in the blood or free floating, the antibody can attack it. So in the first cases, it can attach to the bacteria or virus shown here in orange. And once that binding happens, it's going to signal the phagocytes to come to that area. So you can also see the macrophage coming up, being attracted to the site of injury. So the antibody helps with phagocytic signaling or phagocytosis. It can also clump up the antigen, so it'll bind to the antigen, and that will make it hard for the bacteria to enter cells. So by clumping up pathogens, it prevents the cell entry, and it can also attract or signal complement proteins to come and get rid of the pathogen. The other thing is, if there are toxins made by bacteria and they are in the blood, it can also neutralize those toxins. Speaking of antibodies for the coronavirus, so people who have been exposed to the coronavirus and survived it would have developed some antibodies in their bodies, specifically towards that virus. So in Italy, a large number of people were infected with the coronavirus. They are thinking of testing the people for the antibodies, and if they have antibodies, the right antibodies, then it means that they have beaten the virus and it is a potential marker of immunity. So they're thinking of allowing the people with the right antibodies back to work. Sometimes the antibodies are used as a type of treatment, as in the case for Ebola infections. So you can see in this picture the antibodies, which are the Y-shaped proteins binding to the Ebola virus, which is shown in red. And once they stick to the virus cannot enter the host cell. Therefore, it cannot reproduce and it cannot spread to other cells in the body. Also, by sticking to the virus, the antibodies do help the body to better recognize a virus. So, a virus with antibodies on it will be better recognized than a virus without the antibodies. So, So here's a little bit of research showing you that fermented foods help in boosting immune system. So in this case, the pickled turnip, so basically has a lot of lactic acid bacteria, and so people who were consuming that type of food seem to have many more antibodies towards the flu. The cells involved in the specific immunity are called lymphocytes, and they are specifically the B cells and the T cells. They are also made in the bone marrow, similar to the other blood cells. The B cells, after they are made in the bone marrow, will continue to mature in the bone marrow. By maturation, we mean that they develop their surface receptors in the bone marrow itself. The T cells, after they are made in the bone marrow, will migrate to the thymus where it will finish its maturation. So B cells are responsible for something called humoral immunity. They are the cells that make the antibodies, and T cells are responsible for cell-mediated immunity. In other words, if the pathogen is free floating, the B cells kind of take care of it. And once the pathogen is inside a cell and the cell is infected, then the T cells will get rid of those infected cells. The B cells are involved with antibody mediated immunity because they produce antibodies, And the T cells are involved with cell mediated immunity. There are two types of T cells, cytotoxic and helper T cells. And in the next few slides, we will see how each of these cells respond to infection. It's important to realize that we have several different types of B cells that can recognize different types of pathogens. The problem is we only have a few of each of those types of B cells. So the first time we encounter a pathogen, you know, even though we have a few B cells that can recognize that pathogen, we don't have enough of that, and so we cannot make antibodies quickly, and that is why we fall sick the first time we encounter a new pathogen. Eventually, our body will be able to make a lot of antibodies. Um, so, what happens? How does the body respond to the initial encounter with the pathogen? So, when the pathogen enters our body, the specific type of B cell can recognize that specific pathogen and bind to it. That binding will activate the B cell to undergo rapid cell division, and it makes two groups of cells. It makes a group called the effector B cells and another group called the memory B cells. The effector B cells do not live long. They make the antibodies and they secrete the antibodies. So they are the ones specifically making antibodies, the effector B cells or the plasma B cells. The memory B cells remain in our body, they are long lasting, and they are kept there in case we encounter the same pathogen down the road. So, on a secondary exposure um, to the same pathogen, the memory B cells will be quickly activated and they will quickly make a lot of effector B cells and also some memory B cells. So, comparing the primary response and the secondary response to the pathogen, so you can see the primary response is how our body reacts on the first encounter with the pathogen. You can see that it doesn't make a lot of antibodies and it takes a long time to make the antibodies. But the secondary response is quicker. The second time we are exposed to the virus, you can see we make the antibodies quicker and we make a whole lot of antibodies. So, this is the importance of vaccination. So, vaccination is the first time you will be exposed to the pathogen, and since your body already has the memory B cells, the second time you encounter that same pathogen, your body can protect you from the pathogen by quickly making a lot of antibodies. So, what do you think about antivenom? It is given to patients of snake bite. Is it a type of passive type of immunity? And the answer is yes. So true. When you receive antibodies from another person, it's called passive immunity. So, for example, when antibodies are passed from the mother to the fetus through the placenta, or when you get antibody injections when you travel abroad. That is called passive immunity. You are not making the antibodies, but you're receiving the antibodies. Active immunity is when your own body makes the antibodies. For example, when you're naturally exposed to the pathogen or through vaccination. So when you make the antibodies, that's called active immunity. So in this case, you know, the survivors of Ebola virus infections, their plasma was transfused to people who were infected with the Ebola virus. So, would that be a type of passive or active immunity? It would be passive, right? Because um, the you know the antibodies developed by the Ebola survivors were extracted from their blood, and it was used to treat the new patients. So the new infectious cases, the patients were not making their own antibodies. They would eventually, but for quicker survival, um, you know, they were given the antibodies from previous survivors, and it was an effective treatment in the case of the Ebola infections. So now how does the T cell work? We looked at how B cell responds. So the T cells, the surface receptors on the T cells cannot recognize the virus and bacteria in the blood. But once it enters the cells, then those infected cells can be recognized by the T cells. So that's why we say that the T cells provide cell mediated immunity. So it what happens is, so once the infected cells sends out signals, then the T cells get activated, and the T cells also make the effector cells and memory cells. And remember, we have two types of T cells. There's the cytotoxic T cell and the helper T cells. So both groups will be made, and in each of those, you will have effector and memory T cells. How does the cytotoxic T cells work? They are also called killer T cells. They're one and the same. So they make a protein called perforins, which can make holes in the cell membrane, in the membranes of the infected cells. And then, of course, water gets in and the cell ruptures. Or they can promote apoptosis. They will actually activate the enzymes responsible for apoptosis or cell death. The helper T cells actually are very important because they activate all the other type of lymphocytes. So the helper T cells first bind to the antigen presenting cell, which is shown in yellow here. And once that binding happens, then they secrete a protein called interleukin 2, which in turn will stimulate the killer T cells and the B and the T cells. And once the killer T cells are activated, they will go ahead and release the perforins and get rid of infected cells, and the B cells will secrete antibodies. So you can see how important the helper T cell is. It is helping or activating both the killer T cells and the B cells to do their jobs. So now you can write the cells that are involved in the specific immunity or acquired immunity, which are generally called lymphocytes. And we looked at two types. The B cells, you have the plasma B cells and the memory B cells. And in T cells, you have the cytotoxic T cells and helper T cells. That brings us to the end of part 2.